Thanks everybody for coming to another episode of our Sarcoma Sisters podcast. Today we have Dr. Corrie Painter, who has done so many amazing things for the sarcoma world. And we're really happy to have her today and to have her talk a little bit about herself and her story, and then the amazing things she's doing with sarcoma. So welcome, Corrie.
Thanks so much for having me. It's a pleasure to talk with you today, and hello to everybody who's watching this.
Yay, welcome, Dr. Painter. Thank you so much for helping us get started with our technical glitches. We're grateful.
We had a lot of technical difficulties, but we're all BFFs now, so it worked out good. Just a couple. So Corrie, I had read your article in Cell where you kind of told your angiosarcoma story. And in that, you kind of talked about these 3 steps to your life being redefined after your diagnosis. And I was wondering if you could just start out however you want, but telling us a little bit about your diagnosis and then how it, with angiosarcoma, and then how ultimately led to your current position at the Broad Institute.
Yeah, happy to. I guess I'll start with the fact that I was about to defend my PhD in biochemistry when I found a lump in my breast. And I was 36 years old. My kids were 2 and 4 at the time. And I think I blew it off as something not terribly important. I was too young to have cancer. And even the little bit that I did know from reading the literature, that a breast tumor doesn't feel the way that this thing felt because it kind of moved within the tissue. It came on overnight. It didn't seem kind of the same as the stories that I had heard and the things that I could read online. And because I was a scientist, I could read the literature and get a sense for some of the medical jargon and what it meant. And I kind of self-diagnosed myself as like being fine. But I called my OBGYN regardless. And that led to a series of frightening events that ultimately cascaded into this diagnosis of angiosarcoma, which is an incredibly rare and aggressive sarcoma that starts in the inner lining of your blood vessels. So if you think of your blood vessels as like a hose, the very inside of that hose is lined with a layer of cells called endothelial cells. And that's what this cancer starts from, is the endothelial cells. And as a result, it can really pop up anywhere in the body where you would have blood vessels. And in my case, it just happened to end up in the breast, but it can arise, like all sarcomas really, in any part of your body. And so the diagnosis itself, I think relative to some solid tumors that are more well known and characterized, it took a long time for them to definitively diagnose me. But relative to sarcomas was relatively quick. And I think that highlights part of the disparities that we encounter when we're dealing with rare cancers, and that the diagnosis itself for a sarcoma can often take so long that a cancer can, grow and spread inside of the body while we're trying to figure out what's going on. In my case, again, like I have a PhD, I'm treated at Harvard. I've got if you're gonna have the best resources and background to tackle something like this, I definitely had those things going for me. And it still took about 5 months, 11 biopsies, a surgery, and, in order for them to give me an actual diagnosis. And through this, it was, I was first, unbeknownst to me, diagnosed with angiosarcoma, though my doctor didn't tell me. I was being treated by a breast oncologist, and he sent my cytology. So they did one fine needle aspiration. They stuck a needle into the middle of the tumor and drew out blood and put it on a slide, looked at it under a microscope, and the pathologist saw one spindle cell. So one cell that was not supposed to be in breast tissue, and that pathologist said it could be angiosarcoma. But because it's so rare, 300 people a year in the United States get this, that the oncologist, feeding the dog, sorry, world of COVID, the oncologist absolutely could not wrap his brain around that actually being the case, and really thought it was something else. So didn't tell me that was the case. And then scheduled me for a series of different biopsies that then escalated into it being angiosarcoma, transferring my care to Harvard at that point, Dana-Farber, because the local community hospital really didn't have any experience and was trying to do right by me. and rightfully sent me to a place that did have experience, which, by the way, highly recommend as the course of action for anybody being treated with a sarcoma, that you have to be seen at a high volume sarcoma center. And they in turn worked up several more core needle biopsies, which were all indeterminate. They could not see the hallmarks of a cancer in any of those core needle biopsies. And so they went in to do a full resection and a lumpectomy. And when I woke up from that lumpectomy, my doctor, who saved my life, my surgeon, said to me, he said, "I've never seen anything that looked like that was not malignant." And so I went ahead and gave you a partial mastectomy to try and increase the chances that I would have clean margins. And sure enough, a week later, I got a call back saying that it came back as a 3-centimeter intermediate grade angiosarcoma. And despite the fact that he did achieve clear margins, the nature of a sarcoma is to grow in these kind of like tendrils. They don't grow like, a solid tumor, carcinoma that might be like a ball. It's more kind of like less defined edges, and they can put like individual cells that can creep out into the tissue. And as a result, my surgeon felt very strongly that it would benefit me and potentially save my life if I went in and had another much more drastic surgery. And so I went in 2 weeks later and had a full radical mastectomy. They took the muscle underlying the breast, and that was 10 years ago, and I'm still alive.
Yay. That is such an amazing story.
That is an amazing story.
Diagnosis, and you asked like, how did I get from there, like all the way to the brood? But like, so,
Yeah.
Yeah, go ahead.
Oh, so I was going to say, just for the people that are listening, I mean, if you're listening, not looking to the video, Lisa and I are kind of doing like the fist pump, clap and stuff. So can you tell the listeners a little bit why 10 years out from an angiosarcoma diagnosis is a air fist pumping sort of situation?
Yeah, it's a very, unfortunately, very aggressive cancer. It's so rare that, and this is going to be the same for really any of the sarcomas that are on the rare side. And even I'm doing air quotes for those listening, the common sarcomas. There's such little data around the clinical outcomes of people with these diseases that it's hard to get like an actual accurate statistic. And so when I was diagnosed with this, and I was looking in the literature for all the known information that scientists and doctors had ever published on angiosarcoma, I found numbers that were terrifying. It was anything from you have between a 5% and a 30% chance of surviving 5 years with this. And again, that doesn't mean you're cured. It means you're still alive at 5 years. And so, I mean, and that's a big wide margin, 5 to 30%. And even if you take the best case scenario, that's still, you have a 70% chance of dying from this, no matter what, within 5 years. And then who knows after that. And so to be alive 10 years after, and I have no evidence of disease right now, knocking on wood, wherever it is, it is I guess, kind of something that goes against the statistics and the odds. And so, the line from the Hunger Games, may the odds be forever in your favor, I think is something those of us with sarcomas take very seriously.
Yeah, when I was reading your paper in Cell that Lisa had mentioned, a line stuck out in there for me, which I think might be a good segue into what you're doing now in the sarcoma community. But you said as a scientist, you went into the literature, you looked at the data, you saw these horrific survival numbers. And I think, and I'm going to probably misquote it because I don't have it pulled up, but you said, at that point, I decided I had to think about how to teach my 2 and my 4-year-old how to live without me. And I'm just like, oh my gosh.
I know, it just gives me chills. I
know. I'm like, oh gosh, I can't deal with it. So obviously you're still here, which is phenomenal, 10 years out, and your children still have you. But how did those thoughts and that realization and looking at the data, how did that get you to where you are now, where you are very much a phenomenal mom, very much a phenomenal researcher, and a force to be reckoned with? Like, how did that all parlay into what you're doing now? And tell us what you're doing now.
Thank you. Yeah, I will say that those were by far the darkest moments. And I think it was one of those things where I did not want anybody else to ever have to look at their kids and have those same thoughts. And so I was going to use whatever time I had left in order to put some, I like to think of it as like trying to get like the yellow brick road laid out in front of you so that you can finally get to Oz and like figure out what's going on. And at that point, there was absolutely nothing. There wasn't even bricks. We didn't even know what bricks were, you know? And so we had to literally find the mortar and find, you know, some straw and like create a matrix that we thought even could look like a brick. Like that's how far back from anything that could look like a road was for us at that point with this super rare cancer that nobody knew anything about. And so I thought, well, you know what, if I, even if it doesn't go in the right direction, even if it goes like in the opposite direction, at least we'll have that, at least we'll have a direction and we'll be able to like be on our feet and we'll be moving and people will see us and they can help guide us in a better direction. And so my whole life became that, trying to build the infrastructure necessary in order to get that direction so that we can, you know, so that other people could then carry the torch, because I was not thinking it would be me. And so I think step one was really kind of partnering with people in the angiosarcoma community. And that came about as a result of this woman, Lauren Ryan, who I wrote about in that Cell paper, who she was a stay-at-home mom. You know, she had the exact same diagnosis as me, but had it a year before I did, which was a breast angiosarcoma. And she just had so much gumption. She's like, I'm not going to let this thing take me down without taking it with me. And so she had the ideas to start a nonprofit. She was going to raise awareness. Her goal was to raise $1,000 and give it to a foundation so that they would cure angiosarcoma. And I just absolutely applauded those efforts. And then after I applauded those efforts, totally broke her heart when I said, that's not going to do anything. Because she had like, she thought she figured it out, right? And she did. You know, she was absolutely on the right track. But unless you're in the sciences and you understand what the infrastructure really looks like, you know, $1,000 is not gonna, it doesn't do anything, you know, and unfortunately it does for those of you who want to donate. The collection of all of the money together absolutely does. I'm not trying to suggest that,
Right.
but like like trying to cure a cancer with that money wasn't going to do anything. And so it really took her by surprise when I leveled kind of the expectations of what you can do with like different pots of money. And at the same time, you know, after I think both of us came to that reckoning, we both realized, you know what, it doesn't matter. It doesn't matter if it doesn't really cure it, because again, just like making the goal, getting this whole thing started so that there would be direction became kind of like the end game for us both. And so we went ahead and just went full force into fundraising. And, you know, she took care of the, all of the business of starting a nonprofit and doing the business and like the IRS and taxes, all that kind of stuff that I don't know anything about. And I was doing all of the networking in the science world. And so I was trying to get people to, I was trying to like say, okay, if we had like a little bucket of money, would you two work together? And, you know, you have the cell line and you have the, you know, whatever capabilities of doing these different experiments. What if you built something together? And that became kind of my my operating mechanism. Instead of having RFAs and traditional granting mechanisms, it's such a small world. I could just like individually reach out to people that I found were kind of related to this field and try to entice them to work together. And in that way, try to make more bang for my buck. And so in the background of what we were doing, and in the background of still being a graduate student, my PI at the time was willing to let me stay in the lab and not graduate, because we all thought I was going to die. And, you know, who wants to like go through the process of dying and like changing insurance and all that kind of stuff at the same time. So I stayed in my grad lab. I ended up defending my thesis a year later because I was in this effort to like synthesize different research groups, finding that folks were just like, oh, poor little patient, but they weren't seeing me as a scientist. And so I was like, well, I have to defend my thesis. So maybe I won't even tell them that I have this. And so I switched who I was in the eyes of the folks I was reaching out to, and became Corrie scientist. All the while, you know, in the background of this, my team at Dana-Farber watched this whole thing. You know what I mean? They watched me go from Corrie the patient to defending the thesis to being the advocate to being a scientist. And fast forward like all the way to now, and we just co-published a paper in Nature Medicine, which is pretty cool. Anyway, so going back in time though, it was really like this desire to get like this world of advocacy together. And at the time I had since graduated, taken on a postdoc in cancer immunology, and that's a whole story in and of itself, but suffice it to say, this was before it hit the mainstream. And I got a grant through the Cancer Research Institute to study melanoma. I personally thought this is like mesenchymal, close sort of related cancer, but fundable and super aggressive. And melanoma, if I could help a melanoma patient, great. And it's sort of related to sarcomas. And 100 years ago, the original kind of father of cancer immunology was, made these observations in sarcoma patients. So all those things tethered together for me to study cancer immunology. Went really well, had the ability to move on to have my own lab, and really thought hard about this notion of advocacy versus being a principal investigator, and weighing where could I make more of an impact. If I have my own lab and I publish one paper at a time, no matter how great that discovery is, I don't think I could be at a place where I could be paradigm shifting in a way that would really impact patients. Whereas I looked at that versus what I was doing as an advocate, where I could fund multiple labs, where I could get lots of PIs together and have them kind of approach science in a little bit of a different way. Really, fundamental to the work I was doing with them was, I will give you money, but you have to make your data open source. Or you have to, like if you make a model, you have to share it and show me how you're going to share it. Really kind of like encouraging people to like think beyond that one paper discovery that they were going to make and how they're going to change the field, so that folks could have resources, information, and data to help a lot of other people. So at that point, at that, this critical period where I was like trying to consider, where am I going with my career now that I'm living, there was this opportunity at the Broad Institute of MIT and Harvard, and they were looking for somebody who had background in cancer research, that understood the perspective of a cancer patient, and that understood what the world of like cancer advocacy. And they wanted this person to come in and help them kind of build a brand new infrastructure that would include patients in the process of designing and developing studies that could generate data that would be freely available to the rest of the biomedical community. And the point was just to kind of like lift all the boats at the same time, so that people didn't have to reinvent the wheel. You didn't have to go and do all of the kind of like unsexy infrastructural build to get data and, accrue the patients and then make your discovery and then kind of silo that off and use that paper to either, advance your own kind of like clinical agenda and or grantsmanship or any of that stuff, but rather put this out there so that everybody in academia and industry could use it in order to make discoveries, use it to, get rationale for designing clinical trials, and ultimately help as many patients as possible all at the same time.
Yeah, it was like it was made exactly for you, this
It was
position.
crazy. I saw the job description. My mom was visiting from out of town, and I, I was like, should I be a PI? Should I do this? Should I do that? Maybe, and I was like, maybe I'll just get like a normal science, like whatever, like a as normal 9-to-5 science job as
Right.
you know what I mean? So that I could still do the advocacy. And that was really my objective of even looking into this space. And I came across this job description, and the one thing I left out, which I wrote in that cell piece, was that one of the things in the job description they were looking for was a risk taker. And I had been a whitewater raft guide. I packed parachutes, I met my husband skydiving. Like, I love to live on the edge a little bit. And so I was like, yeah, I mean, it was literally like they made this job description based on my life. And I printed it out and I ran downstairs and I read it to my husband and my mom, and they're like, come on. No, that's not a thing. So
That's so amazing.
yeah, it was really meant to be.
So what year? So you got diagnosed in 2010, and then what year did you run down with the job description?
2015.
So 5 years later. So you hit that 5-year mark at that point.
Yeah. Yep.
Where you were alive with no evidence of disease, and you found this perfect job description. And then tell us what that job description, where that's at now, and if I remember correctly, it started out with breast cancer, but that's not where it ended, and that's not where it will end. So can you tell us
Yeah,
a little
it
bit
was,
about what's
I
going
will,
on?
I want to talk about that just for a second. It was for metastatic breast cancer, and it was really put together by Nikhil Wagle, who's a medical oncologist at the Dana-Farber, treating metastatic breast cancer patients. And he was working very closely with the leadership of the Broad Institute and in the cancer program. So like Eric Lander and Todd Golub and several people in that in the leadership of the Broad, thinking about this for years and years. And the thought was to launch this in metastatic breast cancer. And to be honest, when I saw this, I was thrilled, but I was a little nervous that I was not going to be able to really connect with metastatic breast cancer patients, because I had always felt on the outside of the breast cancer world more generally. I always felt like I didn't belong, though I had a lumpectomy and a mastectomy and was a bald woman, and you know what I mean, like had a cancer in my breast, was not a breast cancer patient. And so I was nervous about whether or not people with metastatic breast cancer would be like, she's an interloper, she doesn't belong here, you know, she's not one of us. And it gave me pause. And it was the one thing that it was nervous about. And to be honest, at this point, I never really anticipated or expected to be able to do projects like these in angiosarcomas or sarcomas. And honestly, it was my intention to really just focus on metastatic breast cancer, because I figured what we could do here is we could help people with metastatic breast cancer who are, you know, one of the ways I talk about this is they're dying. Doesn't matter if you have a lot of resources dedicated to your particular type of cancer. If at the end of the day, you're going to die just the same death as anybody else with cancer, they need better resources and they need to live as well. And I thought, well, if we could bring this about and help metastatic breast cancer patients, great. And we'll build this infrastructure. And if it works well, it should be able to be leveraged for other cancers. And that would be great if that ever happens down the road. But mostly what I want to do, and especially after I started working on it and I got to know and be friends with some of these patients, I just wanted to help that patient population. And it just was even, it wasn't even really kind of a flicker in my eyes until way into it that we would do this in my cancer or in other sarcomas. So we did launch in metastatic breast cancer. It took us, you know, I started really in January of 2015, and then we launched it in October of that year. And we built that project with several patients that have metastatic breast cancer, who became, you know, they became part of the study staff, really. You know, they were so intimately familiar with every aspect of that project that when we launched, and we had nothing to point to, so there was no trust in the greater community, like we want to, you know, sequence your data and like make it freely available. Of course, you know, there was a lot of people that are like, well, who are you? You know, why would I do that? Who are you? And all of the people that we had been working with that did hold trust within that community were like, no, they're with us. And I got a lot of, you know, you can trust her, she's one of us, despite the fact that I didn't have breast cancer or metastatic breast cancer. We had this like simpatico like relationship that developed over time. And it really led to, I mean, and I still, you know, several of those people that we worked with so closely have since died, which is heartbreaking. And several are still alive today. And it's just one of those, you know, sad realities when you embark on something like this, that you're gonna build things and fall in love with people left and right. And they're going to pass away, and you have to be aware of that going into it. It's heartbreaking, which is why I get teary-eyed when I talk about these things.
Yeah.
But anyway, so we launched that in October of 2015, and it was really successful. We didn't necessarily have a metric for what success would be. We didn't know if a couple hundred people would sign up, if over years a couple thousand people would sign up. But suffice it to say, we are in 2020, and we've had over 7,000 people join that study.
Wow.
All over from every state. And we've had men and women. We have you know, people from Canada that have signed up and said, you know, count me in, I want to be part of that. We've made our data available, and we continue to make the data available. And now there are over 20 citations that are using that data in order to, you know, drive their own research forward. And so that original hypothesis that we started with, that if we make this available, a lot of people can use it and drive their research forward, is really true. And it's just gaining momentum. And it's fascinating to watch how scientists and doctors are using the data over time as it develops and matures. You know, it's going from more of kind of like we used all publicly available data to, you know, make this discovery, to we searched the Metastatic Breast Cancer Project data and found this, and then we carried on all of these studies, and lo and behold, it's true. And so even watching the way the data is used and changed in the citations has been really interesting. So about a year in, because of the success of this, we decided we wanted to try to apply this to another cancer. And what better cancer than something that's exceedingly rare, that you would never get enough of any individual patients at one institution to do meaningful science with, and that really could collect information from people that are decentralized. Then, you know, that the perfect cancer to study is angiosarcoma, if you really think of it through that lens. Like, could we build something in one patient population and point it to another and make equally, you know, impactful data available? And the answer to that was emphatically yes. Yes, we can, you know. And so we built the angiosarcoma project, and there we had so many things going for us at that point. First, you know, I was already kind of so deeply embedded within the community, and it's such a tiny community. We all kind of found each other in one Facebook group, and we still are there today. There's not enough of us to start multiple different support groups, you know. And so like somebody will try, and then they'll be like, oh, you can do that, and then also why don't you come over here, because we're all over here, you know. And they'll kind of like collect people up, and we all end up there together. So we already had kind of a deep, you know, connection with each other through this group. And then the more important thing is that the metastatic breast cancer patients totally paved the way. So we had something to point to. And I could say, instead of when we would talk with the metastatic breast cancer patients, it was like, envision this with us, help us like come up with the vision, and help us figure out how we would describe this to somebody, you know, so that they can understand it. And instead of being there, we were like, look what we did. We want to do this with you. And it was a much easier kind of process to build it. So we built it off of kind of the framework of the Metastatic Breast Cancer Project, and worked with the patients in order to face it toward our patient community. And we launched it in 2017. And, you know, the first data set that we released, I think either had 14 or 19 patients in it. And you could see immediately that just what we do is we generate genomic data, and we have a team of abstractors who look at the clinical medical records of patients and pull out information. And then we get patients to tell us about their own experience with angiosarcoma. We take away the readily identifiable information and stitch it all together, and then put it out into the public domain. So we put it in places like bioportal out of Memorial Sloan Kettering, and there, you know, a lot of institutes have their own instance of it. And so but anybody can go, you can go to your smartphone right now, you can go, you know, on the computer, and you could just type in bioportal, and then look for angiosarcoma project, and it's right there. And you don't need password or anything, because this is just de-identified aggregated data. And if you go there, there's like 80 different charts, because we have so much clinical information populating this. But one of the immediate striking things that popped out was that one of the charts talks about tumor mutational burden, and there was this like a normal kind of distribution, you know, like a nice little like camel's hump, and then off to the side there was like one peak going up all by itself. You do not need to be a scientist to know that's weird, you know, that's wonky, that shouldn't be the case. And so we looked at that one peak of patients that had really high tumor mutational burden, which, by the way, has you know, been associated with having a positive response to cancer immunotherapies or checkpoint inhibitors. Looked at that patient population, and lo and behold, all of them had cancer, angiosarcoma, that arose in the head, neck, face, and scalp region. And as I said before, the angiosarcoma can arise anywhere in your body, but the number one place it arises is in the skin, in the cutaneous regions that are sun exposed on white people, older white men. And that was who this cohort was. It was these, you know, people that were having it in that skin and sun exposed portions. Now, up until this point in the literature, there was no mention of sun damage or UV radiation causing angiosarcoma. But our analysts went ahead and did a UV signature on the tumor DNA and found that all of these people that had high tumor mutational burden also had a UV signature. You can literally see the sun writing in the DNA of the tumor, I'm here, you know, and you can tell by the way that the mutations arise that it was caused by UV radiation. And so we were able to make that discovery. And then, you know, that led us to think, well, we know immunotherapies seem to, you know, there is this correlation with people that have high tumor mutational burden. And we have these angiosarcoma patients now who are in that category. I wonder if there are any of these patients who've been treated with immunotherapy, and if so, what kind of response did they have? So we then were able to look at the medical records of these patients, and we found 2 patients that had head and neck face scalp angiosarcoma that were stage 4 and put on pembrolizumab and had extraordinary responses to that therapy. And when at the point that we did the last kind of updated check of their medical records, had been off therapy for 2 years and completely without evidence of disease. So it's just absolutely remarkable. And those things were the big kind of major discoveries that we put into this paper that got published this February in Nature Medicine. And there were several other discoveries and highlights that we were able to pull out as well. But it just, you know, it was remarkable proof of concept that you can build something with patients. It will, if you build it with patients, you know, can have ownership in the process and therefore help establish trust within the community for accrual. It can lead to discoveries that you might not otherwise make. Being able to collect medical records from, you know, in all of our Count Me In studies now, we have several thousand institutes that have each had one patient participating in the project. Imagine trying to design a study where you included data from several thousand medical institutes, including individual, you know, community setting hospitals in rural America. You know, that's data that would otherwise just not be collected and go toward the greater good, but through this mechanism, we can all now learn from. And so I'll stop there. I hope that answered your question.
Yeah, no, that's fantastic. And I think it's as you were kind of telling the story about the head, neck, scalp angiosarcoma and the mutational burden and the sun exposure, it's like your life is weaving back to that early, like maybe I should do melanoma, which also respond very well to immunotherapy and all those things. I think that's fascinating.
Yeah.
A little bit of a nuts and bolts question. So the Metastatic Breast Cancer Project, the Angiosarcoma Project, and now the Osteosarcoma Project, which was your latest launch, I believe, they're all part of this kind of greater collective Count Me In organization, right? It's an organization. So what does that mean for a patient who's diagnosed with either one of those metastatic breast cancer, osteosarcoma, or angiosarcoma? What you talk about them taking part in the study, giving their data, some of the sequencing. What is the nuts and bolts of what that project looks like?
Sure. So I'll start with the projects themselves. So we have the Metastatic Breast Cancer Project, Angiosarcoma Project, Metastatic Prostate Cancer Project, Stomach and Esophageal Cancer Project, and All Comers Brain Cancer Project. So, and tumor. So anybody with a brain cancer or tumor of any subtype can join. And most recently, the Osteosarcoma Project. These are open to anybody in the United States or Canada. And that's the only gating step. If you've been diagnosed, especially now because we were able to engineer state by state workflows for folks that are pediatrics for osteosarcoma to be able to join either with consent or assent, depending on how old they are and what state they're registering from. Basically, if they're interested, they can go to any of the project websites. And I think the easiest way to get there is through our parent website, which is joincountmein.org. And then it links out to the individual projects. So a patient with any of those cancers can click Count Me In. And what will happen is they'll be asked a series of intake questions. And those questions, there's survey questions that we built to better understand a patient's experience with that particular kind of cancer. And there's so much information that patients can provide that helps so much more than we even anticipated when we built these in the first place. And I would like to take just a second and talk about how important that patient reported data is. You know, when we first started designing these projects, we thought, well, we want to ask patients who they are so we have a general sense of like who is joining the projects, and that'll allow us to like build cohorts and move studies forward. But what we underestimated was, first of all, how much patients actually know. If you build the questions, this is the key. If you build the questions with the patients, how high fidelity the data is on the back end. That's the crucial step is like, me and Nick tried to build the questions and they got red marked by all of the patients. Like, we know this, but we would never say it that way. So why don't you try asking it this way? Because this is like this is how humans actually talk. And so, and when we design them with the patients, the concordance rate between what a patient would say and what was in their medical record was like super high. And so that being said, as a result of that high concordance rate, we now had a way to like jumpstart our ability to understand not just like who these patients are, but details about their experience with cancer that would have taken so much time had we waited to get a medical record and do a full abstraction to understand who these patients are. They could just tell us. And so really understanding, I just can't underscore how important that is for us to understand who patients are so that we can report out information connected to their genomics just straight from the patient and their own personal experiences. So they provide that information. We try to pare it down so that it doesn't take too long and is not terribly cumbersome. And we make every single question optional so that if they are kind of done and want to hit submit, that's fine too. And once they do that, then they would be presented with a consent form and a medical release so that we know where they were treated. At that point, that triggers our internal staff to go through a bunch of different steps that lead to us mailing out a saliva collection kit for quote unquote normal or healthy or germline DNA, as well as if they provide consent to allow us to send a sample collection kit for blood, which they can bring to their next blood draw at their doctor for a courtesy draw, or we can work with Quest Diagnostics. And so then it goes back into the patient's hand, that next step. They provide the saliva and or blood back to us, and that triggers our next series of steps, which are to call and get copies of their medical records. And once we get their medical records in, we take a look if they can opt in or not to allow us to collect tissue, archival leftover tissue from procedures they have already had. And our team will then look at what tissues they may have available. We have some rules around, not requesting the most recent biopsy, for example, metastatic biopsy, because we don't want to exhaust a patient's clinical samples. But if there's sufficient sample, we then request a portion of the tumor. And so this is a very small little slice that we would take off of a block of tumor that's embedded in wax, and then we would send that rest of the tumor back to the pathology office that we requested it from. And we take that normal DNA that we got either from the saliva or the blood, and we take the tissue that we got from the tumor, and we'll perform what's called whole exome sequencing on both. And what that is, it's looking at the DNA in the gene, the coding region of the DNA of your normal DNA and the tumor DNA, and then comparing what's different. And those are what we use to make the data that goes into these discoveries that I was talking about that show, well, there's definitely things that are different in the tumor. Well, what are those and which genes, and does that drive a cell to divide faster, or does it make it so that it won't be sensitive to a specific medicine, and so on and so forth. And that's the data that we make available.
And then, like you said, that data is available to anyone who basically, how is it that the scientists and the physicians can get to that data?
Yeah, so there's a couple different ways. There are different levels of data that we have. They're all freely available, but we need to be really careful with data that has any ability to be linked to patient. It's still all anonymous, but when you get to that normal, healthy germline DNA, that has to be protected. You can't just release that out into the public because it would be too easy to trace that back to an individual patient. And so we have that in databases that are kind of protected from the Genomic Data Commons and dbGaP. And these are routinely used by sciences that are in an academic setting. They have to basically apply for it. And then those organizations have, different parameters for, data access. And once they are cleared, then they can use that data and they can study it, but they're tethered to it, and they have to be backed by their institution who will vouch for them, basically, so that bona fide researchers are using that really sensitive data. but then the rest of it, the data that I was talking about, where we made all of our discoveries, is just the individual mutations that are called different in the tumor versus that normal, which that's not identifiable. And then the information from a medical record that's been stripped of identifiers. So for example, like somebody's date of birth would be an identifier. So instead of reporting like, you know, this person that was born in like January of 1947, you know, we would say what their age was relative to when they were diagnosed with cancer. So there's no, you don't know when they were diagnosed with cancer. That's you know, and so everything kind of relative to some arbitrary point of time in the eyes of like somebody looking at the data, but you have all of that same relevant information and can still make discoveries off of it. That is in CBio portal where anybody can download it. And several people have used that to develop clinical trials from. And so that level of detail enough for them to make clinical trials out of is available.
That's incredible. So part of my lab life, which has just is starting to come to an end as I transition into more clinical trial work and whatnot, has been in osteosarcoma, which you have a project on, which has a similar incidence rate to angiosarcoma, right? It's, you know, in that kind of 350 range for kids, because I'm a pediatric oncologist. There's a couple more in the adult range. And I think what you said is so important about what, like the number of papers that came out of the angiosarcoma project. You know, I didn't do research full time. I've always continued to see patients, but just working with a really rare tumor like osteosarcoma, you know, it's taken me years, like 7 years to get samples and do the research and get the information out, which is not still all the way out. And it's been on, you know, 130 blood samples from patients. And that's been an extraordinary amount of effort, an effort worth doing, but that's just how slow these projects can run. And so that's going to equate to probably 2 papers in 7 years. So the fact that you can get 20 papers out from, you know, a project within 5 years, that's such an incredible impact. And I think it's fantastic for these rare subgroups, like the metastatic breast cancer is a subgroup of all of the breast cancer. And then these really, really rare diagnoses like angiosarcoma, osteosarcoma. I mean, the impact is incredible. And so as a physician who takes care of the patients, as a researcher who has struggled at the bench and otherwise, you know, we're just so grateful for the impact that you've made, you and your whole team at the Broad, and all of the patients that have participated in these efforts. It's really incredible. So I don't know, you know, I think sometimes if you watch the nightly news or whatever, it seems like, and especially now with everything that's going on with COVID and this rapid turnaround of data that's coming out, you know, sometimes we just think, well, why haven't we gotten there yet? Like, why haven't we done it? Why is this taking you so damn long, right? And so I think it gives you some like barometer for how long this biomedical research can take, and especially for rare diseases like this. And it's not just cancers either. I mean, we saw a whole explosion with ALS and other things in recent years. So, but I think, I don't even know what the percentage would be, Lisa, but I think there's so much that comes from advocacy. And I think you were far ahead of the curve, and marrying those two things is just, you know, your science background, your patient experience, and your love for trying to make the world better. It has just been incredible. So thank you.
Well, thank you so much.
Yeah, I just have to say the number because it was just popped out of me to be on the page when I was reading the Nature article, was that in 18 months, you had 338 patients registered, and there's only 300 patients diagnosed per year. And we already talked about the not great survival rate. So I was just like, whoa,
Yeah.
that is just so amazing, you know, and just the fact that you mentioned, you know, from the metastatic breast cancer patients, how many papers have already been published, and like you said, the limitless amount of experiments and data and future research this will impact and be impactful for patients. You know, it's definitely, I got my MPH a few years ago, so I was thinking of the upstream effect, and it's like, that is what you are doing, and it's just so amazing.
And I think the other thing that, you know, is so incredible about these Count Me In projects is anyone who's ever done data abstraction from a chart. So for our listeners, that's taking, you know, patient Johnny and getting how old they are, what they got for their therapy, what their response rate was, you know, where they lived, you know, their family history of cancer, all that stuff is extraordinarily tedious and fraught with problems because as physicians, Lisa and I being physicians, sometimes we're not great about documenting that in the chart. And so we know it because we talk to the patient every day and we know that they're doing X, Y, and Z, or this is their background, but it might not actually make it into the chart. And if it doesn't make it into the chart, then there's no researcher in the world that can pull it out because it doesn't exist there. That it exists within the personal history of the patient, which I think is what you were talking about, that is such high fidelity information that it can be missed. Quite honestly, I mean,
Yeah.
I wish there was a better way to make that not true, but that's just the way it is.
Yeah, it is. It's the Wild West. I feel like, you know, med records and
Oh, yeah.
the clinical experience is kind of like the Wild West of medicine right now.
Yeah, that's it. That's incredible. That's incredible.
I did have one other question I wanted to ask you that I saw was part of the osteosarcoma project that hadn't been on the angio one. And I thought it was so cool was that loved ones could contribute if their loved one had passed away from osteosarcoma. And I just wanted to know if you could just speak a little bit to how you all came up with that and then what that process is, because I didn't click on it. I just saw that it existed and I thought that was super cool.
Yeah, so we actually built this in the angiosarcoma project, and this is straight out of,
it is there.
Yeah, it's straight out of my role as an advocate.
I love it.
It was this was really born out of the fact that, you know, people in my support group are there oftentimes years after they've lost somebody, and they need closure. They have to have closure, and they have to be able to contribute to somebody else, like just not meeting the same fate that their loved one met. And there's no way for most people to do that. And I think that's why a lot of charities start and why a lot of people give money, because there is no blueprint for here's how you handle the incredible, intense grief of losing somebody that you love to an awful disease that doesn't have a cure. There's like not one right way to go about it. And there's no real infrastructure for folks to feel like they've been able to just, you know, take a step that gives them enough space to breathe. And over the years, folks have just been like, I'm cleaning out my husband's office. I absolutely cannot throw his medical record away, but I can't have it here and I don't know what to do. I'm sending it to you. And I'm like, okay, you know what I mean? Like, okay, absolutely, you know, send it to me. But there's not been a way to leverage that to help others. It's most, you know what I mean? And so when we were building the angiosarcoma project, I thought, well, what if we gave that space to people? And then maybe that could be kind of like the first step in us maybe obtaining archived tissue from people who passed away from the disease. You know, a lot of centers have to hold on to tissue for X amount of years, even if they does pass away. And so we developed a survey for loved ones to tell us about the experience that they had. And what I did not really count on was just, again, like these are people who held the hand of somebody who died from this awful rare disease and was with them 24/7, oftentimes the point of contact with that medical team. And they hold and possess so much information, and they have never been leveraged in biomedical research before. And so I'll just use like an anecdote that when we started collecting data in the angiosarcoma project, we asked, you know, just basic information. When was your loved one diagnosed? What age were they? Where did it start? Where did it spread? You know, those kinds of things. And we ended up, one of my colleagues, Michael Dunphy, made a Sankey plot, which I always call a snaky plot because, you know, that happens, out of the original site that these loved ones reported the cancer arose and then where it spread to. And we saw immediately after he took that data from the loved ones, there was over 100 surveys that were filled out, that people that had cardiac angiosarcoma looked like it. One of the main places that the Mets would go to is the brain, but that wasn't ever in any piece of literature anywhere. And so we didn't publish it, but it was just like, whoa, look at that. We did present it at ASCO. And it was, you know, it was a plot in one of our posters and presentations. And lo and behold, last year, there was a publication, and it was a multi-institution, retrospective analysis of cardiac angiosarcoma. And their big discovery was that there's Mets to the brain. and we knew that just literally from asking loved ones about the experience that they had watching somebody die from this disease. And so it just, again, is like, a clue, like it's just another thing that we can learn. And I think when you have these ultra rare diseases like angiosarcoma and osteosarcoma, no stone can be left unturned. And with the osteosarcoma, like just talking with folks, like there's this woman, Teresa Beach, who's lost her son Daniel to the disease. And she is, literally a space engineer and knows, a lot about research in general. And so she has taken it upon herself to like listen to the community. And if there's a hint, she chases it. And so from our perspective, hi, Wendy's child. From our perspective, when we talk with people in the community, they're going to provide us information like that we can then parlay into questions that would never be on the radar if we didn't talk with people and didn't like really talk with the loved ones who really have this lived experience with somebody who died from the disease. So in a nutshell, loved ones.
Yeah, totally. Oh, you are talking on mute, Wendy.
Correct. I'm on mute. I just can't get over how valuable and how impactful your personal experience has been with kind of marrying all of this together, because, I've heard over and over and over from my patients' families after their loved one has passed away, like, I just want people to remember my child. I just want them to continue to matter. And how do I make that happen? And it's not the end of it, for sure. It's not the only way to make, to help with that closure, but it's such a large piece of it. And it's so therapeutic. It's that marrying of, humanity and science together. And how do we bring it all together to leverage good for the greater good? And I think I'm just like so in awe of how much like your experience and how perfect you were for this project. Like, this is just so incredible.
I really appreciate that. Thank you.
Well, I know we want to be respectful of your time, but I did have one more burning question.
Okay.
It's so scientific, which is, how did you come up with the turtle mascot for angiosarcoma?
That was Lauren. So Lauren, who started that Facebook group, yeah, she was, I think she, after her diagnosis, went for a long walk on the beach, and there was this turtle slowly making its way back out into the ocean, and or making its way, after laying eggs, making its way like back out to the ocean. And it was just one of those things where sometimes, it's the slow and steady that's going to ultimately get there and win the race.
Yeah, I love it.
And my last burning question. So I left out a bunch of the projects you have ongoing. You said there's one, like an all-encompassing tumor protocol. Is there any plans for the future for specific disease projects upcoming that you can tell us about? I don't want you to share any like trade secrets.
Yeah, no, we don't have anything that's like that's fleshed out enough to really talk about, but we are absolutely, every day we consider like, what's next, what are we going to do? And we're always talking with folks and talking with advocates and scientists and funders in order to figure out like what is next. So what I can say for sure is there's definitely a next chapter. And we will, the ultimate goal is to be able to do this in all cancers. And also to build resources so that folks that are studying other diseases will have, like software capabilities. So our software was developed as an open source software so that people could more easily and readily develop patient partnered research projects. And they are being leveraged already from folks outside of the cancer world to to be able to build these types of projects. And so, the whole goal is to ultimately like build as much data, build it in as many places as we can, and then help both inspire and provide resources for other folks who want to do this in different spaces.
That's excellent. This is so cool.
Thanks.
Yeah. We are so grateful for all of your time, and grateful that you're on the earth right now at the same time as us, and you're making such a huge impact on the world and on cancer, and of course on our little sarcoma world.
Appreciate it. Thanks.